Bioinfocongress V Dr. Özgen Deniz The Rules of Transposable Elements in Cancer. Dr. Özgen Deniz, lisans eğitimini moleküler biyoloji ve genetik alanında Orta Doğu Teknik Üniversitesi'nde, yüksek lisans eğitimini moleküler ve hücresel biyoloji alanında Ruprecht Karl Üniversitesi'nde tamamlamıştır. 2014 yılında Barcelona Üniversitesi'nde Profesör Modesto Orozco ile doktora eğitimini tamamlamış ve araştırmaları sırasında Maya'da nükleozom konumlandırması üzerine odaklanmıştır. Doktor Deniz, doktora sonrası eğitimi kapsamında erken gelişim ve kanserde transpozonların epigenetik düzenlemesini araştırmak için Londra Queen Mary Üniversitesi'nde Dr. Miguel Branco'nun grubuna katılmıştır. Bu çalışmalar neticesinde memeli gelişiminde transpozonları düzenleyen epigenetik yoldaki iki anahtar enzimin yeni rollerini keşfetmiştir. Ayrıca lösemide onkolojik güçlendirici olarak hareket edebilen ve kanser hücresel fenotipine katkıda bulunan transpozonların ilk örneklerini sunmuştur. 2021 yılında Imperial ve AstraZeneca tarafından finanse edilen kendi araştırma programını kurmak için Imperial College London'da araştırma görevlisi olarak görev yapmaya başlamıştır. Dr. Deniz 2022 yılında Cancer Research UK Kariyer Geliştirme Bursu ile ödüllendirilmiştir ve şu an London Queen Mary Üniversitesi Bars Kanser Enstitüsü'ndeki Hemato-Onkoloji Merkezi'nde araştırmalarına devam etmektedir.
Sayın Doktor Özgen Deniz ve değerli katılımcılarımız, Bioinfocongress V etkinliğimize hepiniz hoş geldiniz. Değerli hocamız bizlere bugün The Roles of Transposable Elements in Cancer konulu bir konuşma gerçekleştirecektir. Konuşma bitiminde ekibimizin hazırladığı ve katılımcılarımızdan gelen sorulardan oluşan soru-cevap bölümümüz olacaktır.
Çok teşekkürler Senem. Sunum paylaşmadan önce izin verirseniz ilk bir dakikayı Türkçe ile başlamak istiyorum. Sonra İngilizce geçiş yapacağım bilimden bahsederken. Öncelikle hepimizin Cumhuriyet Bayramı kutlu ve mutlu olsun. Atatürk'ün bize bıraktığı bu eğitim sistemini yetiştirdiği bilim insanlarından biri olarak bugün Cumhuriyetimizin 100. yılında bu konuşmayı yapmak gerçekten çok gurur verici. Ve beni davet ettiğiniz için çok teşekkür ederim. Sizin gibi bilimseverlerle online'da olsa buluşmak gerçekten, tanışmak gerçekten çok mutluluk verici bana. Umarım bu konuşmam sırasında hepimizin öğreteceği şeylerden bahsedebilirim. Nice bilim dolu yüzyıllar diyerek İngilizce geçiş yapıyorum ve slaytlarımı paylaşıyorum. Ve bir soru olsa lütfen söyleyin. Evet, şu anda slaytlarımı görebiliyor musunuz?
Şu an görebiliyoruz, evet. Dilerseniz sunumunuza başlayabilirsiniz.
Tamam, şimdi İngilizceye geçiş yapıyorum. Çok teşekkürler. Thank you again for the invite, and today I will talk about the roles of transposable elements in cancer. So I'm gonna touch the unusual part of the genome. These are not the non-coding part of the genome. And I hope that I can manage to convince you that this part of our genome is actually very functional and very important. So as mentioned in the introduction, I'm actually a new group leader, and our lab has been established like a year ago. So everything that I'm going to present here are quite preliminary data. So transposable elements. So what are transposons? Transposons are DNA segments that can move from one part to the other part of the genome, therefore also called jumping genes. They have been first discovered by Barbara McClintock in 1950s from her studies in corn chromosomes. When they were discovered, she actually called them controlling elements because they control the gene expression. And here, actually, you see different colors of kernel that are due to the transposons. So they modify expression of the coloring genes and therefore impact the whole phenotype. And 40 years after their discovery in 1983, Barbara McClintock was awarded a Nobel Prize for her fascinating studies on transposable elements. And today, we know that transposons are found in almost all organisms, including eukaryotes and prokaryotes. They are typically in large numbers, but still their abundance varies between species. For example, they can be as low as 1.6%, such as some prokaryotes, or they can be as high as 90% of some species, such as in some plants. So what about human genome? In human genome, we know that the protein coding part, so the genes, compose a very minority of our genome, only 2%, where most of the cancer studies, in fact, most of our biological studies are focused on this tiny portion. On the other hand, transposable elements compose almost half, even more than half of our genome. So therefore, the vast majority of our human genome is composed of these repetitive transposable elements. Based on their DNA structure and replication mechanisms, they are divided into different subclasses. For example, there are very few DNA transposons that uses cut and paste mechanisms. The second class, retrotransposons, use copy and paste mechanisms. This is actually the reason why they can multiply in a very quick time within the genomes. These elements require an RNA intermediate to transpose from one part to the other part of the genome. This includes lines, signs, and LTRs. So just to show you the abundance of these elements in our genome, I'm just going to show you a snapshot of a region, that is TP53 gene. This is one of the most important gene in the cancer studies. This is a single gene with all the exons here and introns are indicated. All these gray, blackish boxes are transposable elements. So you can appreciate how abundant they are in our genome. In fact, we have 4.5 million of elements in our genome. So here the biggest question is, with such parasitic burden, how do hosts cope with this amount in our genome? So there are two reasons for that. I'm going to explain the second one later in a couple of slides. But the first one is because most of these elements are not active anymore. So they cannot move from one part to the other part of the genome. In fact, only non-LTRs, these are mostly lines and a little bit of signs, they continue to proliferate. But this is still at a very low rate. And in fact, less than 0.05% of these elements are active today. So since they are not active anymore, these actually immobile elements, the jumping genes, have been considered as junk DNA in our genome. So the research committee thought for a very long time, these elements are just sitting in our genome without any function. But large-scale genomic studies such as ENCODE and Human Genome Project that actually started looking at their relevance and functions changed our view about them from junk DNA to treasure DNA. So we started discovering their functional relevance in our human genome. And today I will give you a couple of examples of their potential roles in the genome in the context of cancer. So the second reason that how the genomes cope with such parasitic burden is because these elements are tightly regulated by various epigenetic mechanisms. For example, DNA methylation is one of the most abundant system that repress transposable elements. So this DNA methylation is is an epigenetic mark that directly affects DNA modification, and this allows the suppression of any epigenetically activated regions. Here I'm just showing DNA methylation in these black lollipops on a full-length LTRs. So here, these full-length LTRs, which actually derive from ancient retroviruses that have been infected in our germline like many years ago, they actually derive from the viruses, and like pretty much like viruses, they compose of coding region that encodes for GAG, POL, and N proteins, and regulatory regions indicated here with these blue lines, which are required for their own transposition, pretty much like how viruses replicate. But in the course of evolution, mostly throughout recombination mechanisms, majority of these elements lost their coding regions. Therefore, they cannot jump anymore, as I explained earlier. But they still maintain their intact regulatory region indicated here with these blue lines. So we and others demonstrated that some of these elements escape these repressive marks, repressive epigenetic regulations, and can be activated epigenetically and recruit host-specific transcription factors. Some of these elements can be also activated transcriptionally and even translationally with potential consequences in the host genome. So, for example, majority of the transcription factor binding sites are embedded within transposable elements. So this plot shows us percentage of transcription factor binding sites in two cell lines. One of them is human myeloid cancer cell line, and the other is a mouse cancer cell line. And you can see the occupancy of different transcription factors that overlap within transposons. For example, 30% of oncogenic TAL1 transcription factors is within transposable elements. On the other hand, in mouse, 40% of CTCF, this is a 3D chromatin regulator, 40% of the binding site of these transcription factors are within transposable elements. So these elements where the transcription factor are bound are highly associated with active chromatin regions. So there are various examples how transposons can actually impact transcription networks and in turn the cellular phenotype. Just to give you one example, this MER41 is an ancient LTR. Here this acts as an enhancer. So upon infection and interference secretion, this particular element is activated as enhancer, promote expression of AIM2 gene, which contributes to innate immune response. So in turn, this element actually contribute to trigger innate immune response, contribute to the cellular phenotype in the human genome. So transposon activity is perhaps most overt in cancer cells, where the genome undergoes epigenetic dysregulation. So all of these repressive marks, epigenetic repressive marks, are disrupted in the cancer genome. In fact, we demonstrated earlier that certain transposon families can be epigenetically activated, and by recruiting relevant transcription factors, they can act as oncogenic transcription factors, promoting oncogene expression, triggering cellular proliferation, and therefore increasing cellular fitness. But these genome-wide epigenetic dysregulations can also impact same or different transposon families that could potentially also regulate tumor suppressor genes. And as a result of this, there will be decrease in cellular fitness. And thirdly, as I thought, some of these elements are derived from viruses. Once they are activated, they can generate unusual cytosolic nucleic acids, such as double-stranded RNAs, and recognize as exogenous viral infection, triggering innate immunity and leading to immunogenic cancer cell death. So given all this opposing and different impact of transposons in the cellular fitness, in the lab, we are trying to actually decipher the roles of transposons in this highly heterogeneous population of cancer cells. So we have many questions, open questions, that we want to answer. For example, whether the same transposon can act as oncogenic and immunogenic, or are they different families, or within the same families but different elements, or they are completely regulated by different mechanisms, or somehow there's like a balance system in the cancer cells that can decide which transposon role is more important for cancer cell, therefore this is used. So to answer all of these questions, or at least some of these questions, we are using acute myeloid leukemia, briefly AML, as a model system. AML is a highly aggressive blood cancer that largely occurs in older adults. One of its important properties, it is biological diverse disease in terms of its genetics, epigenetics, and clinical outcome. So 5-year overall survival rate varies greatly among patients within different subtypes depending on the age, but in general this is quite low. So 5-year overall survival rate is 20%. This is a lot worse in the older patients. So if you look at here the survival curve, the purple and the green ones, only very tiny portion of the patients, like this is 2-3%, can survive after 5 years. So there's clearly unmet clinical need here for the AML. So another property of AML, that's why we are so interested, is that it is an epigenetically driven disease. So the majority of the patients have a mutation in one of the epigenetic modifiers. These are usually targeting the repressive parts. That will lead to removal of the repressive epigenetic layers. And also its epigenome is highly dysregulated. This actually provides a fertile ground for transposon activation. Moreover, its heterogeneous clonality and its hierarchical structure will allow us to model the co-option of transposable elements as AML evolves through these 3 different stages: proleukemic hematopoietic stem cells, to leukemic stem cells, and finally AML. So today I'm going to talk about 2 little stories that we started generating data in the group. As I said, we are newly established lab, so these are all preliminary data. I will highly appreciate if you don't post any of these results. So the first story is about oncogenic roles of transposable elements. So I told you that some transposons can be epigenetically activated. And to identify these epigenetically activated transposons, we first profile chromatin accessibility sites using DNA-seq assays. So this chromatin accessibility gives us open chromatin regions and therefore epigenetically activated sites. So we have performed these experiments in the cell lines. We also use public available data from 2 different AML consortium. And we also included healthy myeloid cells in our analysis. And you can see the results of our complicated analysis here in this heat map, which shows DNA-seq enrichment, which yellow shows high enrichment. So there is a high chromatin opening, chromatin activation here, and blue is low enrichment. This shows repressed and closed chromatin structure. So with our analysis, we identify 6 transposon families that have open chromatin structures in AML cell lines and AML patient cells, but completely repressed and closed chromatin structures. So they are epigenetically repressed in healthy myeloid blood cells. Interestingly, all of the 6 transposon families we identify all belong to the Altair class, that are Altair 2C, 2B, 13A, 5HS, 5B, and 12C. Here you can see in detail the DNA-seq signals or chromatin accessibility sites within each family. So if you're not familiar with such heat maps, I'm just going to go through one of them. So for example, Altair 2B, I forgot to say at the beginning. So these families that we identify transposon as families based on their evolutionary age and based on the signal similarities. So within a family, there are various number of copies. For example, for Altair 2B, we have 320 copies in our genome that is scattered in different parts of the genome. And all of these 320 copies are very similar in terms of DNA sequences. So here, each horizontal line represents a single copy that belongs to the Altair 2B family that is located in different parts of the genome. And you can see the heat map of all of these Altair 2B elements belong to the Altair 2B family. So darker colors indicate high DNA-seq signal. So there's a chromatin opening, open chromatin structure. So here you can appreciate that except Altair 12C, there's a very consistent DNA-seq signal in the majority of these Altair families that we identify. So there is like certain chromatin opening in a specific site of the transposon families that is indicative of transcription factor binding events actually. In fact, when we analyze all of the ChIP-seq data from a myeloid cancer cells from ENCODE consortium, we found that all of these transposon families are bound by multiple transcription factors, the majority of which are essential for normal hematopoiesis, so normal blood formation, but also involved in the etiology of AML. I here only showed some of them as schematic representation, which includes oncogenic MYC and TAL1 transcription factors, or key regulator of normal hematopoiesis, such as SPA1 and Icaros transcription factors. So I don't have much time to show you that, but in addition to what we show, these transposable families, they have high chromatin accessibility, so there's chromatin opening, and they can be bound by transcription factors. We also show that these elements are enriched by enhancer-specific chromatin mark, showing that these elements have potential to act as enhancers. So next, we ask whether these elements can directly modulate nearby gene expression and activate it as active enhancers. To answer this question, we perform both genetic and epigenetic manipulation of these elements and look at their nearby gene expression, whether their nearby genes are changing any expression. So firstly, genetic editing, deletion of candidate transposons. Here, when we remove the complete LTR element, one of the LTR elements that act as enhancer, this leads to significant reduction of the nearby genes in both heterozygous deletion and homozygous deletion, showing that this element acts as an active enhancer, regulating the expression of the nearby gene. We also took advantage of their sequence similarities. As I said, within a family, different copies have very similar DNA sequences. So taking advantage of these, using only a few guide RNAs, like four guide RNAs, we can target entire family and suppress hundreds of LTR copies at the same time using this epigenetic editing. So using this strategy, this is a CRISPR-I, this is called CRISPR-I. So here we recruit repressor crab protein to our targets. So our targets are hundreds of LTR elements. We can suppress them, which lead to downregulation of their interacting genes, but also importantly, reduction of cellular growth in the leukemia cells. So the black one are our control cells, and the red ones are the cells where we target these transposons. So this is also very important, showing that these families we identify can act as oncogenic enhancers, and they are actually essential for cellular growth in leukemia. But the picture is not that simple. AML is a highly heterogeneous disease, as I told you, and it's in terms of its genetics, epigenetics, and clinical outcome. The clinical outcome vary greatly among patients, even within the same molecular subtype. So here, this is the overall survival curve of patients that belong to the all same molecular subtype. That is the largest AML subtype that has NPM1 mutation. So while some patients have very bad prognosis, some patients do much better in clinics. Very interestingly, this cellular heterogeneity is reflected in the LTR chromatin profile. So this is the ataxic profile, or similar to DNA-seq. This is again looking at chromatin accessibility sites, open chromatin region, on two regulatory LTR families. And different patients indicate by different color that all belong to the same molecular subtype. So here you can appreciate the heterogeneity among the patients. While in some patients, this particular LTR family is highly activated, in some of the others, it's completely repressed. So here the question is whether this heterogeneity is pathologically relevant, or some of these LTRs can actually contribute to adverse phenotype seen in patients. To answer these questions, we decided to focus on the largest AML subtype that has NPM1 mutation. This nucleophosphamine mutation in nucleophosphamine gene. NPM1 is a multifunctional phosphoprotein, and one of its main functions is chaperone histone proteins and ribosomal proteins between cytosol and nucleus. When there is a mutation, the protein is mostly located in cytosol and cannot enter to the nucleus. So NPM1 is frequent to occur. The NPM1 mutation occurs 1/3 of the AML patients, so it's a very large subtype, and it also frequently occurs with DM3A and FIL3 mutations. So the patients that have these 3 mutations are called triple mutant patients, triple mutated AML. So this triple mutated AML occurs around 10% of the AML patients. And recently it was demonstrated that triple mutated AML identifies a new subtype with a very bad adverse prognosis. So you can see the survival curve of the triple mutated AML, which is quite low overall survival rate compared to the rest of the AML patients. Interestingly, mutations in both of these genes, FIL3 and DM3A, is associated with enhancer hypomethylation and enhancer activation. So we hypothesize that in the presence of these three mutations, there is an enhancer rewiring, which can lead to activation of certain transposons, which could trigger another layer of transcriptional regulation and trigger oncogene activation, contributing to adverse phenotypes observed in these patients. So to test this hypothesis, we analyze chromatin accessibility sites. So again, open chromatin regions, epigenetically activated sites in our genome, using 3 different AML patient cohorts. These 2 are public available data, and the last one is generated in our group. So we divided the patients into 2 groups as poor risk and favorable risk. Poor risk group patients have triple mutations, NPM1, DM3, and FIL3. And favorable risk patients are the rest of the subtype, NPM1 subtype. So we first wanted to see whether there is a global change in the chromatin activation and inactivation in these 2 patient groups. So when we look at the genome-wide chromatin accessibility sites, we can clearly see that in the blue boxes, which are poor risk patients, there is a clear chromatin accessibility gain comparing to the favorable risk patients. So there is more chromatin activation in the poor risk patient groups. And when we look at our cohort, the data that we generated, that we still have the patient samples to do any functional assays, any further assays, we also observe that this commonly gained chromatin accessibility sites, so poor risk specific open chromatin regions, are enriched by enhancer specific histone marks. So you can see that with this heat map. So we perform histone profiling that is specific to enhancer mark, HTK4 monomethylation and HTK27 acetylation. These are the histone modifications enriched in the enhancers. So these 2 marks will tell us the location of enhancers. And these profiles are done in 3 primary AML cancer cells. And you can appreciate that all of these poor risk specific open chromatin regions are enriched by these 2 marks, showing that there's a clear enhancer gain in these poor risk patient groups. So focusing on the repetitive part of our genome, we reanalyze this data just to minimize any biases that, as these experiments are done in different labs, and the analyses were different. So we reanalyze all of this data and identify transposon families that are common in all of these 3 data sets. And we identified 7 transposon families that are specifically activated in poor risk patient groups comparing to the favorable risk patient groups. So these are the names of the ALTR families, transposon families. Again, all of them belong to the ALTR class. And these are the copy numbers of these elements. So for example, ALTR2B has 353 copies in our genome. On the other hand, ALTR12C has 2,765 copies in our genome. That's scattered in different parts of the genome. Again, remember here, every family has different copy numbers, but there's very high sequence similarities. Interestingly, when we look at their evolutionary age, we can see clearly that except ALTR41B, all of these elements are primate specific. So they have been inserted into our genome relatively recently. But relatively recently, by recently, I mean this is still 50, 40 million years ago. Just to give specific examples, these plots demonstrate active copy numbers of the given transposon families in 2 different patient groups, poor risk and favorable risk. Again, poor risk groups are blue boxes and favorable risk groups are black boxes. You can clearly see that in all of these 3 regulatory ALTR families that we identify, there is a clear chromatin accessibility gain. So there's clearly more epigenetic activation on these regulatory ALTRs in poor risk patients compared to the favorable risk patients. So when we look at the transcription factor motifs, we found that these elements are enriched by binding sites of oncogenic HOXA9 and MACE1 transcription factors, or oncogenic HLF and MYC transcription factors, as well as key regulators of hematopoiesis and 3D chromatin regulator CTCF. We also provide some of these transcription factors, such as HOXA9, MACE1, and CTCF, and we also demonstrated that these transcription factors are bound to this regulator LTRs. So this data suggests that these LTR families that we identify are specifically epigenetically activated in poor risk groups, and by recruiting relevant transcription factors, they have potential to regulate nearby gene expression and contribute to transcription networks. In fact, when we look at one of these LTR family, LTR41B, that was shown to be bound by CTCF and SPI1, we can see that in the active LTR41B copy, that we define this with chromatin accessibility sites, these active copies are also enriched by enhancer-specific marks. And when we look at the gene expression, LTR41B vicinity genes, so the genes that are close to LTR41B or interacting with LTR41B are more upregulated. They're upregulated in poor risk patient groups compared to favorable risk patient groups, showing that LTR41B elements contribute to transcription networks in this poor risk AML subgroup. Just to give you a specific example so that you can visualize rather than looking at the plots. So this is LTR41B element is inserted or it's found in the intronic region of mTERF1. This is a mitochondrial transcription termination factor 1. It's highly expressed in AML patient, and its high expression is associated with poor prognosis in AML. So the higher this gene is expressed, the worse outcome of the patient that we have. So when we look at the promoter region of this gene, you can see that the ataxic profile, so this is open chromatin regions, the profile is quite similar in all of these patients, poor risk patients and favorable risk patients. There are some tiny differences, but still there is a very clear signal, so there's chromatin opening in the promoter region. But when we look at this LTR41B element that acts as an intronic enhancer here, there is a activation, poor risk group specific activation of this element. So these elements highly activated in poor risk patients. We can judge this by the presence of this peak. So there's a chromatin opening here, but it's completely repressed in favorable risk group. So this peak is gone. So this means that the chromatin is closed. This LTR41B element is completely repressed in these favorable risk patient groups. And in line with these chromatin profiles, the relevant gene is highly expressed in poor risk patients compared to the favorable risk patients. So far, our preliminary data of part 1 demonstrated that certain transposon families that all belong to the LTR class are epigenetically activated, and we think that by recruiting oncogenic transcription factors, they can contribute to transcriptional networks in these poor risk patients. So to test their functionality and determine their role in the cellular phenotype, we are at the moment setting up culture conditions and CRISPR system in primary cells, which are extremely difficult to manipulate and grow and manipulate. So here we are using a co-culture system where we grow primary AML cells, cancer cells taken direct from patients with murine stroma cells indicated with this red. And here you can see the photo of our culture system. These round and nice cells are cancer cells taken from patients, and these blurry ones are stroma cells. So using this co-culture system, we can actually grow these cancer AML cells, patient AML cells, up to 3 to 4 weeks in dishes without changing their morphology and chromatin structure. So here the red is the cell number. So of course they preferred a lot less in the dish in these culture conditions, but here the idea of this co-culture system is to maintain the cancer cells so that we can do our functional assays. And the blue is the stem cell mark, showing that there is not much decrease. So the cells still maintain their cellular morphology in this co-culture system. Importantly, their chromatin structure is also largely maintained in its co-culture system. So this is the ATAC-seq data, the chromatin accessibility sites, open chromatin regions. In the cells that we co-culture for 3.5 weeks here, compared to the cells directly taken from the same patient. So you can see that there's a large overlap, showing that this chromatin structure is largely maintained, which is very important for our experiments, because when we are doing our functional assays, we want to maintain the chromatin structure as close to as possible in vivo, what's happening in patients. An important pilot experiments demonstrate that we can deliver these two vectors that are essential. These are essential for CRISPR experiments to silence transposon families in the patient cells. So we can successfully deliver them with high efficiency, and after 3 weeks, the cancer cells still express the required vectors. This gives us an optimum window to perform our functional assays and to test the importance of these transposon families in ex vivo systems. So to sum up our first part, I demonstrated that certain transposon families, most LTR families, are epigenetically activated in poor risk AML, which are normally repressed in favorable risk patients. And we think that by recruiting, now they have the chromatin accessibility, DNA accessibility, so the chromatin is open, they can recruit oncogenic transcription factors, modulating transcription networks, and contributing to adverse cellular phenotype observed in the patients. So the second part, I will talk about another role of transposons. They have quite multifaceted roles. They are much more complex than the talk that I'm giving here, but I just wanted to mention, as I said, two important roles of transposons in the context of cancer. The second one is immunogenic roles of transposable elements. So I told you that some of these transposons are derived from viruses, and they can trigger an innate immune response. So how this happens is, and there are various studies demonstrated that upon epigenetic treatment, certain transposons can be activated. So there's a broad activation of transposons because you remove the epigenetic layer completely with these epigenetic treatments that can generate cytosolic nucleic acids such as double-stranded RNAs that will bind to the DNA or RNA sensors and activating a cascade of events, which in turn activate interferon 1 and 3, leading to activation of innate immunity, innate immunity, and leading to immunogenic cancer deaths. So this whole process is called viral mimicry because transposon activation mimics viral infection. So the cells think that there is a viral infection and tries to kill the cancer cells as a consequence of activation of transposons. So, but so far, which transposons actually play a major role in triggering innate immunity has been not completely studied. So again, using AML, we wanted to study which transposon has more roles and whether we can manipulate their expression without using epigenetic drugs to trigger innate immunity, whether they can be combined with immunotherapies for certain patients that can be more beneficial. So one of the problems of epigenetic therapies in AML, so there are quite a few that are already in clinics, but their consequences or the clinical trials were not as how we expected. So some patients respond, less than half of the patients respond, but only like 10% of them they don't relapse. And this is quite patient specific, and we cannot predict which patients can respond to these therapies. So therefore, it's very important to understand molecular mechanism of epigenetic therapies. So in the lab, we wanted to first define which epigenetic therapies could trigger the robust immunogenic activation, and then find out which transposon is responsible for that. And instead of this global impact of epigenetic drugs, we can just activate these transposons and trigger immune activation and kill the cancer cells. So this part has been done with two very talented students in the lab. They first want to try different epigenetic therapies to see which one can lead to robust immune activation. They also combine different epigenetic therapies because they impact different layers of epigenome. So here we use three main class of epigenetic therapies. One of them is targeting DNA methyltransferases that affects DNA methylation pathway. These are destabine and azacitidine. These are DNA hypomethylating agents. They lead to decrease of DNA methylation. The second one is histone deacetylase inhibitors. This is a panobinostat. So basically, this leads to more histone acetylation, opening of the chromatin activation, and again leading to transposon activation. The third one is histone methyltransferase that targets the repressive histone marks such as H3K9 trimethylation or H3K27 trimethylation. So overall, all of the epigenetic therapies basically target repressive marks, epigenetic marks that will lead to chromatin activation overall. I'm just showing you the, and of course we have done these experiments in various cancer cell lines, but I'm just going to show you the results from one cancer cell line, and I will explain the reason why. And we tried different concentrations, different timing, and we found that decitabine that targets DNA methylation pathway, and panobinostat that is a HTAC inhibitor leading to more histone acetylation, gives us the most robust activation of interferon stimulating gene 15. So this is, I show that this transposon activation leads to cascade of events, and this is one of the final stages of this cascade, activation of interferon stimulating gene 15. So we observed that upon the destabine and panobinostat treatment, the ISG15 activation was a lot more compared to the other treatments. And this is also the case when we look at immunogenic transposons. MER21C is one of the LTR elements located in chromosome 12. This is a single LTR, 400-500 base pair, and this was shown to be an immunogenic transposon. So we use this as a control. Again, destabine and panobinostat leads to activation of these elements. So we decided to focus on these two epigenetic drugs that target two different epigenetic layers. We were also wondering whether, since the destabine hits the DNA methylation pathway and panobinostat hits the histone acetylation pathway, we thought perhaps combining these two drugs might have a broader impact and broader activation of transposons. And in fact, you can see that when we combine these treatments, we have a lot more activation of all the immunogenic genes and immunogenic transposons. This result is in line with what was observed before. So the use of demethylating agents such as destabine and HTAC inhibitors such as panobinostat we used here in combination have been previously shown to have synergistic effects in cancer cells. And we think, and our results suggest that this is true immunogenic activation of innate immune response. And in fact, when we look at the RNA-seq data, we can see that upon treatment of HTAC inhibitor, destabine, and in combination even more, there is a activation of all of these genes that are playing a role in this innate immune system. This is interferon regulatory factors, viral defense genes, immune response signals that are all activated upon treatment with these two drugs or in combination. To look at these results in an unbiased way, we also look at the most upregulated biological processes upon treatment with these two different drugs. And again, the hits were among the immunogenic pathways, such as cytokine production, immune system processes, response to external stimuli, response to viruses. So these two drugs clearly leads to activation of innate immunity. And in line with our hypothesis, we think that this is true activation of transposons. So in our RNA-seq data, we can also get the information of the transposon transcript from the total RNA-seq. So we look at the transposon activation upon treatment of these two epigenetic treatments in the cancer cell lines. And we've seen that this is just showing ERB class. So this is just subset of transposons, but you can see that majority of them are highly upregulated upon treatment either with the sabin or HDAC inhibitor. So here the red ones are upregulated and blue purplish ones are downregulated. And from this heat map, we can also see that while some transposons are activated regardless of the treatment, some of them show really treatment-specific activation, such as this element or this element. So among these activated transposons, we can also see alter-12C that was part of my part 1 talk, that we also identified this as oncogenic transposons. So here, this is acting as immunogenic transposons. So when we look at the whole T, the 50% of our genome, we can see activation of all the classes of transposons upon treatment. While there are some commonalities between treatments and combination, there are also treatment-specific activation of transposons. So I told you that this result is only from one cell line, because in other cell lines we tried, they're all AML cell lines. We treat them exactly with the same drug, the same concentration. We didn't see any of the activation of transposons, neither immunogenic, neither the genes that are playing a role in innate immunity. So this was quite puzzling. Why did we just see these results in one cell line? Interesting, this cell line has the M3A mutation. So we have kind of a hypothesis of that, and I'm trying to explain this. And this was also postulated by other researchers in the field. So regardless of the epigenetic therapies, there are some priming events, such as cancer-initiating mutations. In our case, this is the M3A mutation. Or there's epigenetic reprogramming. So there's activation of certain transposon that can generate double-stranded RNA. So this goes, the level goes to up to here. But this is still subletal. This is not enough to trigger interferon response. In this case, epigenetic therapies act as a booster event. So that leads to even more activation of transposons, so broader activation of transposons, increasing the double-stranded RNA levels or other transposon products, passing this threshold. That's now there's enough in the cell, in the cytoplasm, double-stranded RNAs to trigger interferon response and wild mimic activation. So in our case, this event is DMT3A. So DMT3A is a DNA methyltransferase that is responsible for DNA methylation. So normally transposons, as I said at the beginning, they are tightly regulated by epigenetic mechanism. So they're highly DNA methylated. They're highly methylated, and they usually look like that, like black lollipops. When there's a DMT3A mutation, it impacts its catalytic activity. So there's less of DNA methylation. So there's already some activation. This is in line with the priming event that I was telling you. So there is a certain transposon activation. And therefore, the DMT3A mutation here acts as a priming event. And combination with epigenetic treatment, there is now enough activation of transposon leading to interferon response. So if this hypothesis is true, then correction of the DMT3A mutation should lead to the reversal of this activation of viral mimicry or interferon response. So to test that, we use CRISPR-Cas9 system. We corrected the DMT3A mutation is OCML3 cell lines that we use. That the only cell line that we observe this interferon response. And we repeated all the experiments in these two cell lines. And very interestingly, we saw these results. So in the presence of the DMT3A mutation, as I said, there is activation of immunogenic genes, as I showed you before, and activation of immunogenic transposons, as I showed before. But once we correct the DMT3A mutation and treat the cells exactly with the same treatment days and concentration, now their activation is completely gone. So we managed to suppress the activation of this interferon response. Again, this is our RNA-seq results, show exactly the same. In the presence of the DMT3A mutation, the orange ones are upregulated. All the genes involved in this pathway, innate immunity pathway, are upregulated. And once we correct this DMT3A, their upregulation is gone. So they don't change the expression. So we managed to show that upon epigenetic treatment in the presence of DMT3A mutation, there's immune activation. And once we correct this DMTA mutation, these epigenetic treatments don't really lead to immune activation. And we think that this is true transposons. And again, you can look at the transposon activation, and in the presence of DM3A, there is a high activation of transposon. You can see that they're all upregulated. And once we correct this DM3A mutation, the transposons don't change expression anymore, so they are not upregulated. So we are still digging into this data. This is quite interesting for us, but just to show you some couple of examples, I want to show you this ALTER12C because this is a little bit of my favorite transposon family at the moment. That ALTER12C is highly upregulated upon destabine treatment, but this upregulation is much lower when we correct this DMTA mutation. And with HDAC inhibitor, this is the same. This is highly upregulated, and there is no activation of this element when we correct this DM3A mutation. So to sum up this second part, we think that our DM3A mutation acts as a priming event. As I show you that with the DM3A mutation, there's a loss of DNA methylation, activation of certain transposons, but that's not enough to pass the threshold level. So there's the cancer cells still continues to proliferate. They are still happily growing every 24 hours. But once we treat them with epigenetic treatment, either using DNA hypomethylation agents, leading to more loss of DNA methylation, or also taking other repressive mark, histone, the acetyltransferases, for example, that leads to broader activation of transposons, and now that's enough to trigger viral mimicry and interferon response. This actually would explain why some patients respond better to epigenetic treatment than the others. So to conclude my talk so far, I think we have more questions than I started with, but what I show you so far is so transposons have multifaceted activities. Some of them can act as oncogenic regulatory regions, that they promote cancer cellular proliferation, increasing cellular fitness, and some of them can act as immunogenic transposons, leading to activation of innate immunity and cancer cell death. So we think that actually this part of our genome, 50% of our genome, act as rheostat in the cancer cells. So they maintain this fine balance, and these are the ones that actually determine whether they can trigger immune escape or oncogenic events, epigenetic changes, and therefore regulate the cancer cellular phenotype. And with that, I would like to finish my talk by thanking as well our collaborators, founders, and our newly established group. And last, I would like to make an announcement that we have 2 postdoc positions available in the beautiful sunny London. If you heard in the background the rain sounds, this is not true. But so we have 2 positions open, and I'm really happy to take any questions.
Sunumunuz için çok teşekkür ederiz hocam. O halde ekibimizin hazırladığı ve katılımcılarımızdan gelen sorulardan oluşan soru-cevap bölümümüzün ilk sorusuyla başlıyorum.
Tabii ki.
Lineman'ın tümör hücrelerine oksijen girişi veya endojen indüksiyonu kanser tedavisi için yeni bir model olabilir mi?
Oksijen girişi veya endojen indüksiyonu. Türkçe güzel oldu. Evet, yeni bir model olabilir. Biz Lyman üzerine çalışmıyoruz ama Lyman üzerine birçok çalışma var ve endojen indüksiyonuna bakaraktan yapıyorlar ve hatta bir tane Lyman benim bu anlattığım materyallere göre daha farklı. Daha onların protein productları, RNA productları, DNA productları hepsi ayrı işlemlerden, hepsinin hücre üstünde ayrı görevler olabiliyor. O yüzden Lymanlar biraz daha komplike alt yerlere göre ve kesinlikle bir kanser tedavisi için yeni bir model olabilir. Ama bu son çalışmalarda dikkat edilmesi gereken tek şey RNA, DNA productlarının, Lymanlarının hangi hücresel olaya etkisi olup olmadığına bakmakta bir fayda var bu tedavi için kullanmakta. Ama kesinlikle kullanılabilir. Hatta bunun üstüne birçok çalışma var. Bizim alanımız bunu kapsamıyor ama evet var.
Çok teşekkür ederiz yanıtınız için. Sıradaki sorumuz kromatin transposable element düzenlemesinin kalbinde yer alıyor. Ancak kromatin işaretlerinin transposable element susturmadaki rolü ve transposable lokuslarında kromatin işaretlerinin nasıl oluşturulduğu ve sürdürüldüğü hakkında ne kadar bilgiye ulaşabildik?
Çok güzel bir soru. Şimdi doğru. Mesela DNA metilasyonunun hatta evrimleşmesinin transpozonları sadece susturmak için olduğu düşünülüyordu uzun bir süreye kadar. Ya da başka bir tane Krabs finger proteinler var. Bunlar da mesela çok güzel örnekler. Krabs finger proteinler tamamen transpozonların DNA sekanslarına göre evrimleşiyor. Ve mesela yeni bir tanesi çıktığında yeni Krabs finger çıkıyor. Sonra bu transpozon kendini değiştiriyor. Bunu tanımlamaya başlıyor ve bu transpozon tekrar aktif oluyor. Sonra yeni bir tanesi çıkıyor, bunu susturuyor. Yeni bir tanesi çıkıyor, bunu susturuyor. Yani bu konuda birçok çalışma var ama şey konusunda haklısın. Mesela biz hala birçok şey bilmiyoruz. Mesela bizim baktığımız genel transpozonlarda DNA metilasyonu çok önemli diyoruz. Ama hangi kontekst için bunu tamamen bilmiyoruz. Ve en büyük mesela challenge şu anda dediğim gibi biz mesela family level'e bakıyoruz. Birçok kopya bakıyoruz ama her kopya birbirinden farklı olabilir. Bilgi olarak şuna ulaşabiliriz mesela. Bir family mesela Lyme-1'lar bildiğimiz şu mesela kanserde DNA metilasyonları çok az. Ulaşamadığımız bilgi ve hatta gelecekte de ulaşmaya tasarlayabildiğimiz bilgi de Lyme-1'ların çünkü bunlar çok uzun 7-8 KB şeklinde ve hepsinin DNA metilasyonuna bakamıyoruz. Lyme-1'ların hangi kopyalarında acaba DNA metilasyonu yüksek, hangilerinde daha az? Lyme-1'ların hangi alanları promoter region mı, internal region mı, hangileri DNA metilasyonuyla birlikte susturuluyor, hangileri susturulamıyor? O bilgiye ulaşmak için biraz daha teknoloji, yani şu anda o teknolojiye sahibiz de o bilgiye ulaşmak için birazcık daha zamanımız var. Ve bu da dediğim gibi challenge'lardan bir tanesi. Ve şu an bizim ulaştığımız bilgide ve şu ana kadar söylediğim kromatin değişiklikleri de genelde family level'larda geçerli bir bilgidir.
Çok teşekkürler detaylı yanıtınız için. Sıradaki sorumuz kanser hücrelerinde fonksiyonel ve dokuya özgü artırıcı etkiye sahip transposable elementleri tanımlamak günümüzde ne kadar mümkün olmuştur? Prognostik belirteçler olarak ele alınabilirler mi?
Dokuya özgü artırıcı aktiviteye sahipleri bu mesela kanser hücrelerinde evet bu çok güzel bir soru. Biz şu anda bunu yapmaya çalışıyoruz. Çünkü mesela sadece kanser hücresine değil, mesela bütün vücudumuza baktığımızda her bizim dokumuzda, her hücre çeşidimizde başka transpozonlar aktif oluyor. Mesela bir embriyonik stem cell'e baktığınızda ki aktif olan transpozonlarla bir mesela hemopatik stem cell'e baktığınızda aktif olan transpozonlar aynı değil. Doğal olarak mesela bir lung, bir akciğer, karaciğer, bağırsak hücrelerine baktığında bunlar tamamen kanser olmasına da gerek yok. Bunlar da tamamen farklı. Çünkü hücresel bu özellikle mesela epigenetik kromatin değişimleri hücresel ve dokusal olarak da değişiklik gösteriyor değil mi? O yüzden transpozon aktivasyonu dokusal olarak değişiklik gösterecek. Ve mesela bu kanser çalışmalarına baktığımızda ne kadar bazı transpozonlar, yani sayabileceğim 2-3 tane transpozon familya var. Mesela bu yeni Lyme-1 HS, Lyme-1 PA2, 3 gibi bunlar genelde bütün kanser hücrelerinde aktivasyon gösteriyor. AML bunların içinde değil mesela. Ne kadar bir kısmı bütün kanser hücrelerinde aktive gösterse de bizim yine de kanser spesifik dediğimiz aktivasyonlar çok fazla var. Öbür soru neydi? Bu hücresel...
Devamı şöyleydi. Prognostik belirteçler olarak ele alınabilirler mi?
Evet, prognostik belirteçler olarak ele alınabilir. Mesela Lyme-1'ların aktivasyonlarını, mesela Lyme-1'ların dediğim gibi bu protein productlarını tespit etmeye başladılar. Geçen sene bir tane makalede çıkmıştı ve bunu şu an kliniğe taşıdılar. Bunu kanser hücrelerinde şey yapabiliyorlar ve bu prognostik olarak kullanılmaya başlanıyor yani. Ama şey konusunda gerçekten haklısın. Yani fonksiyonel ve dokuya özgü artırıcı etkileri kesinlikle var. Günümüzde daha da mümkün olacak. Biz şu anda elimizdeki bilgiler genelde bu kanser atlaslarına dayalı. Yani şu anda gerçekten öyle kapsamlı bütün bir aynı labdan çıkan ya da bu kolaborasyon bir labdan çıkan bir bilgi yok.
Çok teşekkürler. Çok sağ olun hocam. Sıradaki sorumuz transposable elementlerin kanserdeki rollerini incelerken yaşanan zorluklardan biri olan normal fenotipik özelliklerin aksine, özellikle onkojenik ile ilişkili transposable elementlerin bulunmasının üstesinden gelmek için kapsamlı bir dizi normal ve hastalıklı hücre durumu boyunca oluşturulan kromatin durumlarından genom segmentasyonunun bir atlası yeterli olur mu?
Bir dakika söyleyeyim.
İsterseniz tekrarlayabilirim. Çok uzun bir soruydu.
Evet. Tekrarlarsanız sevinirim.
Transposable elementlerin kanserdeki rollerini incelerken yaşanan zorluklardan biri olan normal fenotipik özelliklerin aksine, özellikle onkojenik ile ilişkili transposable elementlerin bulunmasının üstesinden gelmek için kapsamlı bir dizi normal ve hastalıklı hücre durumu boyunca oluşturulan kromatin durumlarından genom segmentasyonunun bir atlası yeterli olur mu?
Yani şu anda bizim yaptığımız yeterli olur durumunda. Çünkü benim şu anda size gösterdiğimiz hepsi benim normalde kullandığımız genom segmentasyonu. Normal, hani farklı bir şey yapmıyoruz. Ama doğru, mesela şeye dikkat etmek gerekiyor. Ben dedim ya, sadece transpozonların bir kısmı hala hareket ediyor. Yani mesela bendeki transpozonlarla sizdekiler aynı olmayacak. Ben kanser olduğumda ya da hani her kanser hastasının transpozonu birbirine aynı değil. Yani yeri aynı olsa, yani bir yer değişikliği var. İkincisi, yeri aynı olsa bile bazıları bunlar tabii ki de şeye gidiyor. Mesela mutasyona uğruyorlar. Mesela bir tane transkripsyon faktör bağlanacakken bende bir mutasyon oluyor. Bu bağlanmasını engelliyor. Yani ne kadar o transpozon aktivite olsa da burada mutasyon olduğu için bağlanamayacak duruma geliyor. Aslında bu tür çalışmaları onkojenik şeylerini yapmak için, gerçekten bu tanısal ve tıbbi alanda kullanmak için kişiye özel gen diziliminde çıkarılması gerekiyor. Tamamen bu çalışmaların gerçekten faydalı olabilmesi için. Ama tabii bunları her transpoz için söyleyemem. Mesela bazıları, dediğim gibi, yani şöyle bir şey yok yani. Mesela kanserde P53 diyoruz, değil mi? Bütün kanserdeki etkisini neredeyse biliyoruz. Kaç binlerce makale var. Transpozonlar konusunda böyle bir şey diyemiyoruz mesela. Gerçekten hani bir tanesi gerçekten şunu yapar da diyemiyoruz. O yüzden kişi değişikliği var. Bu heterojenite çok önemli. Heterojenite her anlamda hem genetik hem epigenetik. Mesela şey de farklı olabilir. Benim genetiğim tamamen aynı olabilir ama epigenetiğim de farklıdır. Ben de aktive olan, ben aynı kanser hastası olan, aynı kanseri paylaşım başka bir aktivite olmayabilir yani. O yüzden bence evet, uzun sözün kısası gerçekten geçerli tıbbi anlamda kullanmak için. Yeterli, pardon.
Çok sağ olun hocam yanıtınız için. Sıradaki sorumuz, konakçı faktörlerin transposable elementlerle etkileşime girdiği ve transposable elementleri düzenlediği çok sayıda yolun ve bu faktörlerin türler arasında nasıl değiştiğini anlamak için transposable konakçı ilişkisinde nelere dikkat edilmeli?
Şimdi şuradan örnek vereyim. Crab-Zinc finger protein. Bir dakika, düşüneyim bu soruyu. Yani örnek düşünmeye çalışıyorum da. Çünkü hani evet. DNA metilasyondan örnek veriyorum da, DNA metilasyon genelde birçok türde Crab-Zinc finger de genelde primate. Diğer türlerde olduğunu yapmak. Ya mesela şimdi diğer türlerde transpozonlar farklı olsa bile bu genel mekanizmalar tabii ki de hani homoloji, ortolog, onun olup olmasına bakılması gerekiyor konakçılarda, ama mesela transpozonlar tabii ki de türlere göre farklılık gösteriyor. Ama genel mekanizmaları bitkiler hariç, bitkilerde başka şeylerle yönetebiliyor. Ya da mesela Crab-Zinc finger proteinin insanlar özeli var ya da fareye özeli var. Ama yine de genel mekanizma tekrar Crab-Zinc finger protein üzerine. Açıkçası bilmiyorum. Yani Türkçe anlatamayacağım galiba.
Çok teşekkür ederiz hocam.
Kusura bakmayın. İstiyorsanız sonra yanıtlayabilirsem zaman olursa bunlara mail atarsanız öyle bir şekilde
Tabii
yanıtlamaya
hocam,
çalışabilirim.
çok mutlu oluruz. Çok teşekkür ederiz yanıtınız için. Sıradaki sorumuz, kanserle ilişkili kromatin varyantlarını bulmak için araçlara sahip olmak ve bunların tekrarlayan DNA elementleri açısından zenginleştirmek mümkün müdür?
Bunların tekrarlayan DNA elemanları açısından zenginleştirmek mümkün müdür? Soruyu tam anlamadım sanırım. Kromatin varyantlarını bulmak için araçlara sahip olmak ve bunların tekrarlayan DNA elemanları ya transpolanlar açısından zenginleştirmek mümkündür.
İsterseniz sorunun anlamını kaybetmemesi adına katılımcımızdan sorusunu tekrar detaylı bir şekilde bize iletmesini rica edelim. Biz diğer sorumuza devam edelim isterseniz.
Evet.
Tamam, çok teşekkürler. Diğer
Repetitive
sorumuz...
DNA'yı, evet, repetitive DNA'yı anladım onu.
Diğer sorumuza devam edeyim ben izninizle. Genomların özellikle de kanser genomlarının genetik heterojenliği tarafından engellenmektedir. Bu durum kromatin durumlarının analizi için kişiselleştirilmiş genomlara olan ihtiyacı arttırır mı?
Evet, az önce cevaplamaya çalıştığım soru. Evet, bu genetik heterojenliği var, epigenetik heterojenliği var. Ayrıca sadece insandan insana değil, bütün mesela tümördeki genetik ve epigenetik heterojenliği. Yani çok kompleks. Kanser gerçekten çok kompleks. Mesela sadece kişiselleştirilmiş genomlara kesinlikle ihtiyaç var, az önce söylediğim gibi. Çünkü mesela bende olan transpozisyon sizde olmayabilir. Bendeki transpozisyon mutasyon sizde olmayabilir. Ama üstüne üstlük bir tane daha komplike şey var. Mesela kanserin de heterojenitesi çok fazla değil mi? Yani aynı kanserin her tarafı aynı değil. Her genetiği aynı değil. Mesela AML'de klonal heterojenity diye bir şey var. Birçok klondan oluşuyor ve her klonda aynı genetik yapısı yok. O yüzden her hücrede aynı transpozon da aktive olmayacak. Her klonun epigenetiği de farklı. Her hücrenin epigenetiği de farklı. O yüzden kesinlikle kişiselleştirilmiş genomlara ihtiyaç var ve bunun üstüne üstlük de single cell, yani tek hücre analizine ihtiyaç var mesela kanserden olmuş ve biz bunu yapmaya çalışıyoruz. Mesela kanseri sadece aynı farklı insanların analizi değil, aynı insanı alıyorsunuz. Kanserin her hücresine bakıyorsunuz. Hangi hücresine mesela hangi mutasyon var? Bu hangi transpozon aktive edebiliyor? Hangi epigenetik değişiklik var? Bu hangi transpozon aktivitesiyle acaba korelasyon yapabiliyor? Onlara da bakabiliyoruz.
Çok teşekkür ederiz. Çok sağ olun. Sıradaki sorumuz, transposable cis etkili enhancer etkileri antiviral gen ekspresyonu üzerindeki antiviral etkiye nasıl katkıda bulunuyor?
Şimdi şöyle, ben karıştırmak istemedim. Çok boyutlu iki rolü birbirine karıştırmak istemedim ama enhancer, şimdi ilk kısımda enhancer şeyinden bahsettim değil mi? Enhancer aktivitelerinden bahsettim. Bu mesela onkogenleri aktive edebiliyor. Ama bu tabii ki de bir enhancer antiviral bir tane genin yanında da olabilir ve bunu da aktive edebilir. Yani bu immünojenik rolünden kaynaklanıyor ve enhancer cis regülatör rolünden kaynaklanıyor. Değil mi? So bir yolu mesela cis etkili enhancer, AIM2 mesela gösterdiğim örneklerden bir tanesinde, introduction'da gösterdiğim örneklerden bir tanesinde bu AIM2 genin yanındayken onu aktive edebiliyor mesela ve bu da innate immunity'nin aktive olmasını sağlıyor. Bir yolu bu. Cis etkili enhancer etkilerinden. Ama benim ikinci kısımda anlatmaya çalıştığım cis etkisi dışında bunun aktivite olup bunların double strand RNA yaratıp tamamen virüs enfeksiyonu varmış gibi algılatıp hücreye öyle antiviral aktivitesini arttırması.
Çok teşekkür ederiz. Sıradaki sorumuz, viral enfeksiyonun hangi yönü transposable elementlerin baskılanmasını tetikler? Bir virüse yanıt veren ve transposable susturma makinesindeki değişkenliklere aracılık eden spesifik bir konakçı, sensör ve sinyal yönü olabilir mi?
Evet, keşke bunun cevabını bilseydik. Bütün sorunumuz bu. Anlatmaya çalıştığım şey bu zaten. Hani transpozonların bir tane özelliği yok, birçok özelliği var ve biz mesela bir konakçı veya bir mekanizma şu özelliği susturuyor diyemiyoruz. Yani onu sustururken mesela bu onkojenik aktivitesini susturmuş olabiliyor. Viral virüse yanıt veren aktivitesini susturmuş oluyor. Soru tam bu muydu? Onu tekrar okuyup şey yapmaya çalışacağım. bir virüse yanıt veren ve evet, buna spesifik bir sinyal yolu yok. Mesela ben örnek verdim ya, destabin, DNA hypomethylating editing agent ya da HTAG inhibitor ya da birçok metal tree, bu RNA modifikasyonlarına da etki eden. Ya bu sadece gidip buna spesifik T'leri susturmuyor ya da spesifik olarak da bu virüse yanıt veren mekanizmayı susturmuyor. Yani biz aslında anlatmaya çalıştım bu broad, bu geniş kapsamlı tedavilerle mesela biz aslında birçok şey değiştiriyoruz hücrede. Yani tamamen sadece virüse cevap veren mekanizmayı değiştirmiş olmuyoruz. Böyle bir spesifik mekanizma maalesef işte yok, onu bulmaya çalışıyoruz.
Çok teşekkür ederiz hocam. Çok sağ olun. Sıradaki sorumuz, kardeş kromatitler üzerinde yer alan bitişik alo elemanları arasındaki homolog somatik rekombinasyonun lösemilerdeki etkileri nelerdir?
Şimdi bu konuda biz alo üstüne çalışmıyoruz ama alo elemanları, mesela bir tane MLL translocation var lösemide. Bu MLL yani hastaların genelde adult ve childhood'ları, mesela çocuk ya %6'lık bir oranda görülüyor. Ve bu mesela MLL'lerin birçok 100 tane falan partneri var. Buna göre onkojenik özellik gösteriyor. Ve mesela gösterilmiş bir tane makalede, alüler, çünkü birbirine tabii ki dediğiniz gibi homolog somatik rekombinasyonu çok önemli olduğu için bunların aracıyla mesela bazen MLL translocation'larını yönetebiliyor. Yani bu onkojenik genetik olayların başlamasına yardımcı olabiliyor. Ama buna tamamen, yani böyle bir örnek var birkaç tane, ama tamamen mesela bu alüleri tamamen kaldırırsak bu acaba olur mu olmaz mı bir deneyde yapılmadı. Bu sadece korelasyondan da ibaret olabilir ama katkısı, etkileri şu ana kadar böyle gösterildi.
Çok teşekkür ederiz hocam. Bu son sorumuzdu. Cumhuriyetimizin 100. yılını bilimle kutladığımız konferansımızda bizimle olduğunuz için gerçekten çok büyük bir mutluluk duyuyoruz. Atatürk'ün ümidi olan gençlere bu önemli günde değerli vaktinizi ayırdığınız, bize bilgi dolu bir sunum yaptığınız, bütün sorularımızı yanıtladığınız için gerçekten kendi adıma, ekibimiz ve katılımcılarımız adına sizlere çok teşekkür ediyorum.
Ben çok teşekkür ederim. Benim için gerçekten önceden söylediğim gibi bir büyük onurdur 100. yılda böyle bir konuşmayı yapmak ve sizlerle tanışmak ve şunu da eklemek istiyorum, Türkçe olarak soru okumak ve cevap vermek gerçekten ayrı bir zorlayıcılık oldu. Çünkü sanırım 2006 yılından beri bilimi hiç Türkçe konuşmamıştım. O yüzden hani bazı kısımlara gerçekten yapamadıysam kusura bakmayın. Diğer farklı yanıtlayamadığım sorular olsa da maille cevaplamaya devam edeceğim. Ve tekrar çok teşekkür ederim. Çok teşekkür ederim böyle bir organizasyon yaptığınız için. Gerçekten çok önemli. Hani bilim, yani Atatürk'ün bıraktığı en büyük şeylerden biri de ve bunu sürdürmek de bizim görevlerimizden bir tanesi. Çok teşekkürler.
Çok sağ olun hocam. Bizim için çok keyifliydi. Sizlere sağlıklı günler diliyoruz.
İyi günler.